My greetings to everybody, my colleagues. Very glad to take part in the conference. But I must say that I am not a medician. I am not a biologist. I am a physicist. And I'll be talking about aging from the point of view of biochemical physics. Next. Okay. So there are two general concepts, okay, of biological aging. According to the first one, there is a program of aging. Genetic program sets a limit to lifespan. The alternative concept advocates that aging proceeds stochastically. The goal of my presentation is to demonstrate how the theory of reliability of biological systems unites both concepts, the aging program and the aging stochastics. For each device, normal operation acts alternate these random malfunctions or failures. That is well known from engineering. And in engineering, reliability is defined as ability of a device to perform its function for a given time under given conditions. And the same should be accepted in biology. And aging in biology is similar to wear out in engineering. That was written in the first monograph on reliability and aging of biological systems, which was prepared with my participants and issued in Kyiv, the former USSR, now Ukraine, in 1987. And not long ago, about 30 years later than our book, a new wave of analogous research has been spurred under the style of biological robustness. It was the paper, the article of Japan scientist Kitano, Biological Robustness, the title. It was published in Nature Review Genetics in 2004, without any citations of our previous publications. By the way, for robustness, there is no mathematics. For reliability, there is the mathematical methods in engineering, the theory of reliability. Meanwhile, as it was said by Immanuel Kant about 250 years ago, in any partial doctrine of nature, one can find as much genuine science as there is mathematics in this doctrine, and no more. Well, we, in the model of aging, in the mathematical model, or in the reliability theory model of aging, which was suggested in our papers, we thought that any device from biomolecular nanoreactors to regulatory networks, any device, any biological device, operates this limited reliability, genetically limited reliability. For each and every device, normal operation alternate these malfunctions or recurrent failures. Inasmuch as all reliability facilities, among them preventive maintenance, replacement, repair, and redundancy of functional elements, inasmuch as all reliability facilities are genetically limited, stochastic damages in cells accumulate up to the genetically preset threshold dysfunction. That was from my paper of 1981. And the mathematical models, okay. The mathematical models was found upon the three postulates. Okay. Okay, just a moment. All right. So, I beg your pardon. I beg your pardon. This is the problem with my computer. Yes. So there are any biological system is the structure of hierarchical. There's the structures of the highest level of hierarchy, the so-called supervisors. The preposition to the set of initial flaws in these supervisors or longevity assurance structures represents a random sample of the exponential, though truncated, distribution with the density function of this type. The exponent was used as the simplest asymptotic distribution known from the statistical physics of linear polymer chains. And preposition 3, the set preposition, this time longevity assurance structures accumulate stochastic flaws, and the organism has been assumed to perish the moment that any of these longevity assurance structures develops the preset, genetical preset, threshold dysfunction of mind. So, as a result, we get the function for survival, the survival function of this type. And according to this survival function, what's the problem? Okay, yes, we get the mathematical expression for the intensity of mortality. Intensity of mortality increases this time, increases in age as exponential function. And by the way, it is the well-known in gerontology, for gerontology, well-known Gompert's law of mortality. It is known from the middle of 19th century. So our theory explains this mortality, Gompert's mortality law. You see, a linear function. Of course, there are some more complicated situations, but everything can be solved using some more complicated models. Okay, and there is another empiric relation. It's known for placental mammals. There is an empiric relation of lifespan, maximum life with species-specific lifespan, T, and the rate of the basal oxygen consumption, V, and body mass. Lifespan varies in inverse proportion to the specific metabolic rate. That's the so-called Rubner law of mortality. You see, 1883. Another allometric equation relates lifespan, rate of the basal oxygen consumption, and brain mass. This is the equation, and it is follows from this equation: who is clever, that lives longer. Man, but that's for specific lifespans. But from this law of Rubner, from this scaling relation, it follows that breathing is harmful. Why breathing is harmful? The answer to this question was first done by Denham Harman, American chemist, in his paper Aging, a theory based on free radicals and radiation chemistry. The paper was published in 1956. Harman, years, Harman worked in the donor laboratory, radiation laboratory in the University of Berkeley, in Berkeley. And you see, that's the book, Free Radicals and Aging. It was issued in Germany in 1992. There is on the materials of the symposium on free radicals and aging that was organized in Paris by Britton Chance and English Emery. Britton Chance was one of the founders of the modern biophysics. Okay, and Denton Harman was first who tried to increase the lifespan using antioxidants. He worked with antioxidants as a radiation chemist. And you see from this table that antioxidants were used very successfully. For example, in the first work of Harman, the increase of lifespan of mice was about 30%. Mercaptor ethylamine was used by Harman. Then another antioxidant, BHT, butylated hydroxytoluene, or 4-methyl-2,6-D tart butylphenol was used successfully, and so on. But by the way, resveratrol, modern antioxidant, is also very effective. But vitamin E was not effective. And the main source of radicals in organisms, the main sources are mitochondria, cell mitochondria, because mitochondria consume oxygen, use oxygen to reduce the oxygen into water, and the energy of this reaction is used to make ATP. But mitochondria also work are not perfectly reliable. Random malfunctions of the mitochondrial electron transport nanoreactors in the cells are of first importance since they result in the advance of the so-called superoxide radicals, the chemical products of which are reactive. And this radical was discovered in biological system. It was discovered in 1968 or in 1965, 10 years after the first paper of Harman. It was discovered by McCord and Friedrich. And of course, there is the special antioxidant enzyme system against these radicals. But the special enzyme superoxide dismutase, and then catalase, glutathione peroxidase. But this enzyme defense, the reliability of this defense system is also limited. And from our theory, we were able to derive the expression for maximum lifespan. You see, this relation T in this relation is species-specific lifespan potential or maximum lifespan of the person, of the species. V, respiration rate. E, activity of superoxide dismutase. Q probability of the malfunctions in mitochondrial nanoreactors leading to O2 minus occurrence. U or U probability for the free radical failures to provoke functional violations. And coefficient D just index to incorporate other damage factors that are not associated with oxygen-free radicals. So we get this equation on the below. It should be linear dependence between the reciprocal of the species lifespan and the ratio of respiration rate to activity of superside dismutase. And indeed, it is the case. You can see the linear equations, the linear graphs for brain, for liver, and for heart. By the way, experimental data were used from the paper of Cutler, National Institute on Aging, and Thomas et al. It was done in 1980. Nothing more was published after that time. Nothing more. But you see, they measured superside activity in tissues of animals of 13 species, and in men, in tissues of men. You can see the linear equations as was in accordance with the predictions of our model. And for example, if we use the equation for brain, we can estimate from the free coefficient. You can see, oh, okay. The longevity of brain could reach 250 years if the reliability of the superside defense was absolutely perfect. Well, so the template principle of organization of living systems implies that information, structures, genes, or gene networks rank first in the cell hierarchy. What is legitimate structures? They are special groups of genes located in a special group of cells that supervise on preventive maintenance replacement of functional elements, turnover, metabolic turnover in the organism. Probably they are neurons of a hypothalamic region called the suprachiasmatic nucleus. And what radicals do? What is the so how this O2 minus superset, how it works? As a matter of fact, it is not a superset. It's not an oxidant. From chemical point of view, it's a reducing agent. It reduces NAD+, reduction of NAD+. And this way, it influences epigenetic redox dysregulation in systemic regulatory networks by expression of silent information regulator proteins. That's the special NAD plus dependent deacetylases sucranes, which has been discovered recently. And these sucranes, which serve as the regulators of important cell process, including glucose metabolism, insulin secretion, adaptation to the oxidative stress, and so on, including at last autophagic or apoptotic death of the cell. And so we finished the talk about that it was talk how the systems theory of reliability reunite both concepts of agent, program and statistics. But how antioxidant works? Antioxidant trap radicals in vivo? No, they trap radicals in vitro, in solutions. As for in vivo cells and tissues, there are the specialized enzymes, SOD, for example, in the case of O2-. But elementary comparison of the rate constants and concentrations of superoxide dismutase and antioxidants, it shows that there is the difference, the so great difference in reaction activity. You see, superoxide dismutase traps the O2- radicals more quickly than antioxidants. The difference is 10 in 7 orders. It's impossible to improve the situation by the antioxidants if we will compare just chemical properties of antioxidants and superoxide dismutase. Of course, one of the products of superoxide radicals is hydroxyl radical. It's great oxidant. But this oxidant, OH radical, it reacts with any organic molecules with very high constants, 10 in 10, 10 in 12. And thus, actually, nothing can intercept this radical in vivo because too much other molecules in comparison with the quantity of molecules of antioxidants. So the mechanism should be another one. And again, systems reliability theory standpoint of the situation. Synthetic BHT, it was shown in our experimental papers, which were done with my colleagues from in Kiev Institute of Experimental Kiev Institute of Gerontology. Synthetic antioxidant BHT, butylated hydroxytoluene, improves supply of tissues with oxygen, thereby preventing the hypoxia ischemia induced conversion of mitochondria into generators of O2-. Flavonoids, natural antioxidant flavonoids, induce expression of antioxidant enzymes, SOD and catalase in vivo. It was shown in the great papers of Nelson and company. And so antioxidants, the so-called antioxidants, do provide the preventive maintenance against active forms of oxygen via the systemic regulatory networks, including hormonal mechanisms. Another way, the human microbiome, the adult human intestine contains up to 100 million bacteria that are beneficial to our metabolism and health. In the gut alone, as many as 1,000 species bring to the body 100 times as many genes as our own DNA carriers. I cite this, the paper of Lederberg. It was published in Science about 20 years ago. We should think of each host and its parasites as a superorganism. So the microbiota communicates with the central nervous system, possibly through neural, endocrine, and immune pathways, and thereby influences brain functions and behavior. The imagined concept of a microbiota with brain axis suggests that modulation of the gut microbiota may be a tractable strategy for developing novel novel medicine for complex CNS disorders and medicine anti-aging. By the way, it was this, I cite here the paper of Cryan and Bannon in Nature, but This idea was first suggested by Ilya Mechnikov in 1907. So Mechnikov arises. By the way, yesterday it was the anniversary of the Mechnikov birthday. Okay, and now the last slide, the conclusions. So, excuse me, so the aging is, how can be diminished this? Because, okay. No. Oh, no. Так. Oh, thank you. Okay, so aging, reliability conception. The aging program does exist. Each and every biomolecular devices, from enzymes to supervisors, are designed to operate with the limited reliability preset in the genome program, in the genetic program. Okay. Aging occurs as the consequence of the genetically preset deficiency in reliability of the biomolecular constructions. We all are the victims of the genetically preset program. The free radical timer serves as the effective stochastic mechanism of realization of the program of aging. And antioxidant pharmacology increases the system's reliability by hypothalamic neurons and microbiome. And here you can see 3 sites, because the first things on this topic were published in 1981. And recently, this year, it was published the book in Elsevier. You can see the book Anti-Aging Pharmacology. And in this book, there is the paper, there's a chapter written by me and my co-worker, Antioxidant Pharmacology, Anti-Aging, in the book, Anti-Aging Pharmacology. And so, thank you very much for your attention.